It's tough to follow a keynote speaker from Ireland, that's for sure. So I will give you a description of the microbiome, but sort of with a New York approach. And actually, it's interesting in listening to Dr. O'Neill, some of the relationships that I think we'll be able to show, particularly towards the end of this talk, that may link metabolism to the microbiome. These are my disclosures. And so just to go back in time, just over a decade, the nomenclature microbiome was actually first coined by Josh Lederberg, a Nobel Prize winner. You'll recognize Jerry Weissman, perhaps, to his right at Woods Hole. And the human body is a superorganism with an extended genome that includes not only its own cells, but also the fluctuating microbial genome set of bacteria and viruses that share the body space. And as most of you know, when one refers to the microbiome, it's very complex, meaning that there are microbes in all anatomic sites. And the question is, up till recently, we thought we just carried them on us and inside us. And the real question now is, are they influencing our immunity, our metabolism, and contributing to both health and disease? And so whether you're looking within the nose, the mouth, the lungs, et cetera, there are distinct bacteria that colonize us. And the question is, what role did they play? And increasingly, the numbers of cells that people have talked about, which is there are over 1,000 bacterial species and over 1 million genes compared to 23,000 human genes, and apparently 3 pounds or 3 kilos, that is, of bacteria within our gut. And so the question then becomes, are we an extended organism with our own cells and genes combined with the genes of these microbes that we consort with? And what's changed in the last decade really is our ability to measure these microbes with all of the informatics, so that culturing bacteria is an insensitive way to determine what bacteria are present in a different site, particularly anaerobes. And with new technologies, and let me just see, where's the best way to... With new technologies, you can discover the 1,000 variety of bacteria by their genetic imprint through 16S RNA sequencing or whole genome sequencing, shotgun sequencing. What I'm going to show you today is work that we've done to the right side of this slide, which is to say 16S RNA and the determination of the 1,000 microbes that live in this case, in our gingiva and in our gut, and what distinctive properties might occur in diseases like rheumatoid arthritis and perhaps even psoriatic arthritis. And I'll show you data on the gums and also in the fecal content. I won't discuss all of the other diseases. I mean, we'll talk about rheumatoid arthritis and psoriatic arthritis, but the microbiome, as you know, has been at least studied, if not implicated, in a variety of autoimmune diseases and in obesity, Crohn's disease, et cetera, as a way to frame the immune response that is triggering some of the activation, particularly of Th17 cells in some of these diseases. So let's look first at rheumatoid arthritis. And all of you are familiar with the notion of the preclinical phases of rheumatoid arthritis, so that for years, in advance of the development of the disease in genetically susceptible hosts, whether it's a shared epitope or others determined by GWAS studies, in the presence of an environmental trigger, whether it's smoking, hormones, perhaps gingivitis, then an immune response will ensue, characterized by autoantibody production, cytokines measured in the blood, and among those autoantibodies, as you know, are rheumatoid factor and anti-CCP antibodies. And then what one sees is early synovitis, epitope spreading, and the development of clinical rheumatoid arthritis. And obviously, the holy grail right now is to keep moving to the left on this slide to determine A, what the initiating factors are, and B, what are the cytokine and autoantibody profiles that will detect RA before it occurs in order to move therapy theoretically back to the left of the slide. The areas that have been most studied recently with regard to the environmental triggers and the anatomical sites are shown in this slide. And so periodontal disease, you're all familiar with the story of smoking and the anti-CCP antibodies. I'll review briefly. involvement of the lung that Mike Hollers and colleagues has demonstrated in Colorado that there may be preclinical lung disease that may be a site of bacteria and autoantibody inciting. And then I'll show you some data on the gut microbiome. Now, first, periodontal disease. You go back to old textbooks, and we talk about 1914, this goes back to the early 20th century as well, that it's good medical practice to eliminate foci of infection, including tonsils and localized dental infection. And I guess there were enough anecdotal discussions in those years that removing the teeth and abscesses resulted in an improvement clinically of patients with rheumatoid arthritis. And this was in textbooks as late as the 1940s. This quote was, in fact, from the Steinbroker textbook of arthritis. And Jerry Weissman reinvigorated this hypothesis about 10 years ago, and identifying or raising the hypothesis that it was particularly the porphyromonas in the oral microbiome in patients who developed gingivitis that was the immune response that induced ACPA anti-CCP antibodies. So that the notion was that there's an increased prevalence of periodontal disease in rheumatoid arthritis. As many as 50% of people with RA can have identifiable periodontal disease. Antibody titers to P. gingivalis are elevated in people with RA and who have periodontitis. And the linkage in terms of the immunological sequences, as you know, that P. gingivalis is among bacteria that express the peptidylarginine deaminase, that is the PADI, which converts arginine to citrulline and develops this neoantigen of the citrulated peptides to which anti-CCP antibodies are generated. And that there are some reports, however small, that people with rheumatoid arthritis with periodontal disease who have aggressive periodontal cleaning may improve with their RA. And as you know, doxycycline has been shown by Odell and others to ameliorate or attenuate rheumatoid arthritis and is approved as a treatment for RA. And it may be through antimicrobial effects if, in fact, it is an effective treatment. So what we did at NYU in studies led by Jose Scher in our unit was to look at the microbiome, and I'll show you first the oral microbiome and then the gut microbiome in a cohort of patients that came into our clinics and clinical sites at NYU. And we looked at several different groups. We called one NORA, New Onset Rheumatoid Arthritis, and these were patients within 6 months of the diagnosis of RA who had not yet been treated with DMARDs or biologicals, so they had not seen methotrexate, for example. The other population were what we called chronic. They were rheumatoid arthritis patients with a mean onset of disease about 8 years previously, but they had obviously been treated with a variety of DMARDs and biologicals, and then there were healthy controls. And then clinical parameters were assessed. Our dentist at the College of Dentistry did extensive assessment for gingivitis and microbial. And 16 sRNA sequencing was performed on the biofilm. And what we saw was not shocking and was consistent with what is in the literature, that people with rheumatoid arthritis have periodontal disease. But what is interesting when you look at this, if you look at the top line, all of the RA patients studied, only 9% had healthy gingiva, and the majority actually had moderate to severe gingivitis, as shown as assessed by the dentists. What was striking, however, was that new onset RA, people who had not seen methotrexate, who weren't having trouble with dental hygiene as a cause of their gingivitis, when they presented, 60% had severe gingivitis. And just as interesting, I saw a patient yesterday in the office who had 10 years of RA, and the last 5 years have been extensive gingival disease with implants, bone problems, taking of the implants. So this is characteristic of people with RA, particularly if you look. And the question I think that it raises, is this the inciting factor, or might in fact this be part of the disease process? And I just put that out and I'll come back later. People with chronic RA also had gingivitis. Healthy controls only, you can see 56% had healthy gingiva as expected, although one can see severe gingivitis in the so-called healthy control population as well. So what we then looked at is what was the microbiome at the phyla level in people with rheumatoid arthritis, in the healthy, the NORA, and the CRA. And what you can see is in the NORA, there was an increase in the Bacteroides phyla. And at the genus level, this was largely due, here shown in yellow, to the P. gingivalis. But what was important to note is that, as shown on the left, it was not the presence of rheumatoid arthritis that was characterized by P. gingivalis. It was really simply if patients with NORA, the 60% of them had severe gingivitis, they had P. gingivalis. So really, the P. gingivalis was tracking with the presence or absence of periodontal disease. And that's also shown in this slide, which looks at in patients with arthritis, and each box now is an individual patient. This is the percent of their microbiome, of the microbiota that is represented by P. gingivalis. So about 30% of the microbes in these individuals are P. gingivalis. And you can see that patients with all RA and NORA have, at least half of them will have P. gingivalis, but on the other hand, it's not necessary to have rheumatoid arthritis because more than half of the patients did not have P. gingivalis. And this really tracked, as I said earlier, not with whether you had RA or not, but whether you had periodontitis. And that's simply shown again that no PD, no periodontal disease, and PD was the source of the gingivitis. I'm just going to skip over this slide in interest of time. So what is interesting, perhaps, is that when you look at gingival disease and rheumatoid arthritis, you find the common mediators that we find in RA joints, that is the cytokine shown in the center and the other mediators. And to some extent, the histopathology is similar in periodontal disease as it is in rheumatoid synovitis. So I think it's an open question, although I don't say that the notion that gingivitis induces PADI and CCP is still a viable hypothesis. But it also raises the question as to whether the gums are an extra-articular site for the attack of the immune system. And I don't know, frankly, which direction this paradigm is going to end up going. So I want to then switch to some of the data that we looked in the gut. And here we took fecal samples from the same cohort that I showed you in the periodontal disease. And we looked at RA and a little bit more preliminary data in psoriatic arthritis. And here is, I think, a beginning of a connection to Dr. O'Neill's presentation, is that we think about the microbiome. These are the functions of the microbiome. So in the upper left, you see that it's well established that absence microbes, you don't develop a full repertoire of an immune system, so that one needs exposure to these bacteria to have a healthy T and B cell population. On the other hand, as you know, the microbiome in the gut form a function in terms of nutrition, synthesizing essential amino acids and vitamins, and also metabolic, processing plant polysaccharides and developing end products. And the question is, perhaps, does a distinct microbiome change the metabolism in the gut, and does the altered metabolism then activate the immune system in a way that Dr. O'Neill was demonstrating in some of his data? So in this concept, and in the world of probiotics and others, you know, the antibiotics, diet, environmental toxins, and pathogenic bacteria influence our immune system, as sort of diagrammed in this slide. So there are normal commensals that may have no particular role in activating disease, but there are what may be called pathobionts. That is, they're not the typical pathogenic bacteria like Shigella or Klebsiella that we associate with disease, but they are commensals that have the capacity to activate the immune system within the gut. And they regulate in a balance the host immune cells, the resistance, and tolerance based on in this kind of theory. And if you begin to put this model together, what you see is the potential that you have normal commensals and then these potential pathobionts that are interacting with dendritic cells or perhaps activating epithelial cells directly. And then in the lamina propria, in the various immune system that lines particularly the small intestine, having an interaction with our host immune system, particularly in regulating the balance of Treg cells and Th17 cells that Dr. O'Neill referred to. And somehow this interaction is normal homeostasis interacting with commensals and other bacteria in the immune system, but perhaps with the emergence or the exposure to these pathobionts in the genetically susceptible host, a trigger of immune response may occur in some people. So that's the hypothesis that's being explored now, both in rheumatoid arthritis, inflammatory bowel disease, and in particular. And this field of associating bacteria with Th17 activation, actually Dan Lippmann really was among the leaders in making this association, based on a bacteria that was described in Japan, known as the segmented filamentous bacterium. And what you see in this little spaghetti picture here, in green is this segmented filamentous bacteria, or SFB. And in certain animals that carry this, it's sufficient to activate the epithelial lining, which is shown in pink there, and interact with those cells to induce Th17 cells. And that, using the model of the KXBN arthritis, as well as others, he and collaborators were able to show that if you're kept in a germ-free environment, they did not develop arthritis, but if you introduced this single bacterium, it could induce Th17 cells and inflammatory arthritis in this mice. And this scanning EM is very lovely because it basically shows that this bacterium sort of penetrates into the epithelial cell and presumably activates the cell to develop Th7, an immune response. So these data are shown in this slide from the Wu publication. And basically, in germ-free mice, this isolated bacterium was introduced and is shown to the right. If the SFB feces were fed to the mice, you can see the development in the upper line there of an inflammatory arthritis in the KBXN. And that's associated, just shown over here, with the development of Th17 cell population being induced in the context of introducing this single organism. And what's shown to the right in the lower right panel is that if this is the swelling or the degree of arthritis in the control animals, treatment with antibiotics, wiping out this SFB, attenuated the arthritis. So this is a simplified version of a much more complex disease, but the notion here is that there are some bacteria, perhaps Clostridia, that favor Tregs, and the balanced other bacterium, SFB, in the mouse, we haven't been able to identify that in humans, if there's a surrogate for that, but the balance such that you can activate into the lymph node various activation, let's say with the dendritic cell at the epithelial layer, with the Clostridia and Treg cell generation, and then perhaps with pathogenic commensals, activate a T helper cell, and then sort of populate your lamina propria with a balance of Treg and Th17. And then in diseases like rheumatoid arthritis, according to this hypothesis, have a migration of these inflammatory Th17 cells into the joint. So what about patients? Those were the mice model using the SFB as the prototype. So again, we went back to the same population that I showed you before, and we looked at whether or not the entire microbiome was diverse in people with new onset rheumatoid arthritis, and that's shown in this slide. And the answer is the amount of bacteria in the guts was not appreciably different. There are some diseases like Crohn's disease, and I'll show you psoriatic arthritis, where there's a lower level of diversity within the microbiome of the gut. But what was interesting is that when a principal component analysis was performed, patients shown in red who had new onset rheumatoid arthritis clustered with regard to the species of bacteria that were most prevalent in that patient population compared to healthy, shown in blue. And interestingly, they were different from patients who had years of rheumatoid arthritis, who had been treated. And the flora of those patients, as I'll show you, was really not distinguishable from normal population. And that's shown on this slide. And of 1,000 species that could be detected, the striking finding in these data is that there was a single species of Prevotella that was predominant in the patients with rheumatoid arthritis of less than 6 months untreated. and that's shown in these Nora patients. So the abundance of Prevotella was 30%, 25-30% in this population as compared to healthy chronic RA and psoriatic arthritis. And when we looked at individual patients, again, these are individual rectangles, you can see that in the Prevotella, the predominance was significant in the Nora as compared to the other populations. If you look at what is expected in patient populations, there are some literature that depending on your diet, the region that you live, there are 3 predominant what are called enterotypes in some of these studies. I refer you to this Nature article from a couple of years ago. And there are predominant species, the Bacteroides, the Prevotella, and the Ruminococcus. And what this slide shows is that in the Prevotella, which was what we found predominant in the RA patients, typically only 13% of the population studied had that as the predominance, whereas again in the Nora, that was 70 to 80% of the Nora population. So is Prevotella an initiating factor? Is there something about Prevotella that's driving a Th17 response? Is a question raised by the data that I'm showing you. Therefore, another way to look at this, if you look at in blue, the Bacteroides, again, these are the main enterotypes. In the population that we studied, what's shown in orange is the prevalence in individual patients of Prevotella versus Bacteroides, versus the opposite, which would be, as shown to the left, which would be the more predominantly expected species in a population. So the question now is we talked about the Th17 activation, the immune activation, but is there a metabolic effect of having a differential expression of the microbiome, of your microbiota? And this was in the Times recently. You may have seen it. And it basically speaks to how the microbes in our gut will produce carnitine in this regard. And then that can be converted by the liver into a molecule known as TMAO, which is a recently identified risk factor for heart disease. So just to read the article, this was a study done of individuals given red meat, and those who were meat eaters, the bacteria converted this to carnitine and then the liver to TMAO. So the real culprit of red meat in this study was not the cholesterol or the lipids, but it was the chemical carnitine that is burped out by bacteria in the intestines and converted by the liver. Now, interestingly, vegans did not have the same capacity to generate carnitine based on a different microbiome, at least according to this study. So perhaps here's a link to the prior talk. When we looked at the feces of these patients, the new onset rheumatoids versus the healthy, and looked at short-chain fatty acids, not the immune response, but a metabolic effect, what we could see is that short-chain fatty acids were different, higher in the Nora patients with the Prevotella, including a molecule succinate, acetate, and butyrate. And in listening to Dr. O'Neill, it was fascinating to ask whether some of the metabolic effects of a disturbed microbiome were in fact activating IL-1 in the gut, and that's certainly a hypothesis that based on today's discussion, I think is worth pursuing. There's also things in the gut that at least suggested that the gut is the site of production of some of the autoantibodies. So this is work done in collaboration with Greg Silverman, who's here, and that is in the rheumatoid patients. The ACPA levels are higher in the gut than in healthy controls. And I'll skip that for a second. Oh, I'm sorry. Let me just pause. So there was an intermediate slide that said, what about psoriatic arthritis? So these data, I'm going to show you a couple of slides on the control populations that we looked at in a similar way when we were looking at the RA. And what was shown on the right here is that there was less diversity in the gut microbiome in psoriatics compared to healthies, and rheumatoids. And what was interesting, and this is obviously an eye chart, but these were microbes that were increased in healthy and less in psoriatics. And one of the interesting ones are Ruminococcus, which is less in psoriatic gut, and which has been described to be less in patients with inflammatory bowel disease and Crohn's. So there's a question in this seronegative disease whether there's an altered microbiome there as well. And this just shows here the Ruminococcus being less in psoriatics. And the other thing that was of interest is looking at secretory IgA, and whether, again, in studies done with Greg, the secretory IgA reflecting B-cell activation in the gut was different, and there was elevations in people with psoriatic arthritis in the feces compared to skin psoriasis, that is. So just to summarize where we are, in the mouth, there's moderate to severe periodontal disease in early RA that is characterized when present by P. gingivalis. If you use the microbiome alone, which obviously is not entirely sensitive in terms of prior exposure with regard to presence of antibodies, the presence of P. gingivalis is neither necessary nor sufficient for the development of RA. We don't know if this is due to alternate organisms in the mouth, if that's the site of the environmental exposure or not, or whether other sites such as the lung or gut are the source of an organism that might incite RA. In the gut, there's the single species of Prevotella is present in the majority of new onset DMARD naive. Again, this is limited to the New York cohort that we examined, and we're currently in the process of trying to see if this is reproducible in other patients. This microbiota is associated with increased fecal short-chain fatty acid levels, and that both RA shows increased anti-CCP antibodies, whereas psoriatic arthritis exhibit decreased overall microbiota and a decrease in Ruminococcus, as has been observed in inflammatory bowel disease. So there are more questions raised by these data than obviously answers. They're hypothesis generating, need reproduction. What is the specificity of the T cells induced by these different microbiota? Can you identify epitopes on Prevotella, for example, that are restricting a T cell response, and can you look at the T cells to determine that? How do the T cells induced by the microbiome initiate the downstream autoimmune response? And finally, if there's a treatment down the line, can we manipulate the microbiome to treat inflammatory diseases such as RA, psoriatic arthritis, or lupus? And manipulation might even be things like vaccination if you can find an epitope. And I would add to this slide, does the short-chain fatty acids that we see, are they driving the inflammation in a way that's separate from these immune mechanisms that are on this slide? So I'll end that there are treatments. There's fecal transplants that are now available from your local gastroenterologist, right now for C. difficile, but perhaps in some fashion going forward we will have notion of fecal transplant for the treatment of inflammatory diseases. So I thank you very much for your, I got one more slide. Thank you. And I just want to recognize the work, and Dan Littman on the immunology side and Jose Scher on the rheumatology side.